Some of Earth's deadliest substances became infamous far outside a murder scene. Fluorine gas attacked glass. A poison carried by tiny Colombian frogs could shut down nerves. And in 1994, a laboratory spill covered less skin than a bath towel. Yet 15 days later, the technician was dead from damage that began beneath the surface. The gas that ate glass. Picture yourself in a 19th century chemistry lab, where one element keeps defeating every container built for it. The substance was fluorine, a pale yellow gas so reactive that isolating it became one of chemistry's most dangerous pursuits. Earlier experimenters were burned or poisoned by fluorine compounds, and the failed attempts later inspired the nickname fluorine martyrs. In 1886, Henri Moissan finally isolated elemental fluorine by electrolyzing a fluoride mixture inside a platinum U-tube cooled to about 9 degrees below 0 Fahrenheit, colder than a household freezer.
That cold mattered because the apparatus had to survive long enough for the gas to form. Fluorine grabs electrons from other atoms with exceptional force, which is why it attacks materials that seem chemically tough, including glass under the wrong conditions. Moissan's success earned him the 1906 Nobel Prize in Chemistry. The twist came decades later. In 1938, Roy Plunkett accidentally discovered PTFE, later sold as Teflon, a carbon-fluorine polymer famous for resisting heat and chemical attack. The same element that made laboratory glass a liability became part of one of the most familiar non-stick materials in American kitchens. The war gas that waited. In July 1917, German forces introduced sulfur mustard on the Western Front, adding a new kind of chemical injury to trench warfare. Unlike chlorine, which could announce itself with a visible cloud, sulfur mustard was an oily, blistering agent whose worst effects often arrived only after a delay.
If you crossed a contaminated trench and felt almost normal at first, the next 8 to 24 hours could be the length of an ordinary night's sleep before painful blisters, eye injuries, and breathing problems appeared. The chemical damages DNA, the cell's instruction material, so tissues that constantly renew themselves are hit especially hard. Doctors studying severe mustard casualties during and after the First World War noticed another effect. White blood cell production could collapse. Edward and Helen Crumbar documented that pattern in exposed patients. Researchers later asked whether a related compound could deliberately attack rapidly dividing disease cells. By 1942, physicians at Yale had given nitrogen mustard to a patient with lymphoma, helping launch modern cancer chemotherapy. The irony is brutal but real. A chemical weapon built to disable soldiers helped reveal that the same kind of cellular damage could be redirected against cancer.
The metal poison that evaporates.
Eric Krauss reported the first simple organocadmium compounds in 1917, including a liquid called dimethylcadmium. Cadmium was already known as a toxic metal, but attaching two methyl groups changed how easily the metal could become an inhalation hazard. Dimethylcadmium is volatile and fumes in air, so a spill can send toxic material into the breathing zone far more readily than a solid piece of cadmium. Its boiling point is about 223 degrees Fahrenheit, only 11 degrees above the temperature at which water boils in a kitchen pot. If you entered a poorly ventilated lab after a spill, the danger would not need to look dramatic. Inhaled vapor could expose the lungs, then the kidneys, liver, and nervous system to cadmium. Toxicology studies have found dimethylcadmium more hazardous by inhalation than inorganic cadmium compounds. The liquid is also flammable and reacts with moisture, adding a second problem to the poisoning risk.
Krauss's 1917 synthesis was chemically important, but the later lesson was about delivery. The molecule around cadmium had made the metal far easier to breathe. The poison borrowed from food. Spend an afternoon with an expedition in Western Colombia, and you can see why early researchers assumed the frogs themselves manufactured their chemical defense. Hunters used skin secretions from Phyllobates frogs on blow darts, and scientists isolated the key toxin, batrachotoxin, in the mid-1960s. The molecule locks sodium channels in nerve and muscle cells in the open position, turning a brief electrical signal into a continuous one. A frog only a few inches long can carry a striking amount of toxin on its skin. The surprise came from captivity. Frogs raised without their wild diet lose the batrachotoxins, showing that they do not make the compounds from scratch. Researchers now think the animals sequester them from food, but the exact dietary source in Colombian frogs is still not proven. In 2004, scientists found batrachotoxins in chorazine beetles eaten by toxic birds in New Guinea and proposed related beetles as one possible source in South America. The twist crossed an ocean. The same toxin family also appears in New Guinea birds, suggesting that very different animals can acquire the same chemical defense from what they eat. The metal that blinds to reveal. In Cambridge in 1803, Smithson Tennant identified osmium while studying the dark residue left after platinum ore was dissolved. One of its most dangerous compounds is osmium tetroxide, a pale solid that releases toxic vapor even below its melting point. The contrast is strange. A 1-inch cube of osmium metal would weigh about 13 ounces, yet its tetroxide can leave the solid phase and enter the air at room temperature. If you stood near an unsealed sample, the first warning could be tearing and eye irritation.
Osmium tetroxide is a strong oxidizer that reacts readily with unsaturated lipids in biological tissue. In the eye, exposure can damage the cornea and cause severe visual disturbances. Concentrated contact can produce deep burns and possible blindness. The same reaction that makes it dangerous also made it useful. When osmium tetroxide reacts with lipids, reduced osmium products create dark, electron-dense deposits that preserve and highlight membranes. By the 1950s, electron microscopists were using it as a fixative in stain, allowing cell membranes to stand out with far greater contrast. A compound notorious for damaging eyes became a tool for seeing structures that ordinary light microscopes could not resolve. The acid that skips the pain. A small paleontology laboratory in Perth became the site of a fatal hydrofluoric acid accident in October 1994. A technician spilled concentrated HF across his thighs while working alone.
The burn covered about 9% of his body, less skin than a full bath towel. Yet the danger reached far beyond the visible injury. Imagine rinsing immediately and still not knowing that fluoride ions are moving through tissue beneath the surface. HF can penetrate deeply and bind calcium and magnesium, minerals the heart and nerves need to function normally. The technician did wash with water, but investigators found that his makeshift hose delivered only a weak flow, contaminated clothing stayed in place, and calcium gluconate gel was not available. Those details matter. Water irrigation itself is standard first aid. The failure was inadequate decontamination, not the fact that water was used. His condition worsened, his right leg was amputated a week later, and he died 15 days after the spill. The countermeasure explains the chemistry.
Calcium gluconate gives the fluoride another source of calcium to bind before enough is pulled from the body to trigger life-threatening systemic effects. The fire that burns sand. Sand usually smothers a fire. Chlorine trifluoride can ignite it. Cold War propulsion researchers examined this fiercely reactive oxidizer because it could ignite many fuels without a spark. If you watched a 1950s test from behind reinforced glass, the liquid below about 53 degrees Fahrenheit could turn to gas as the equipment warmed, while the compound attacked materials normally considered fire-resistant. Chlorine trifluoride reacts violently with water and can ignite cloth, wood, glass, asbestos, and even sand. Rocket chemist John D. Clark later made its reputation famous in his memoir about liquid propellants, describing a substance that ignited on contact with an extraordinary range of materials without an external spark.
The storage trick was passivation, a protective skin formed when the chemical reacts with certain metals. Steel, copper, or nickel can develop a thin fluoride layer that blocks further attack, but if that coating is damaged, the underlying metal is exposed again. Military and rocket programs explored the compound because its chemistry was so aggressive. That same aggression helped kill its appeal. An oxidizer that could ignite almost anything also made leaks, handling errors, and damaged containers exceptionally hard to manage. The venom that became pain medicine. In 1982, researchers isolated a small venom peptide from the fish-hunting cone snail, Conus magus. The molecule, later called ziconotide in its synthetic drug form, blocks a type of calcium channel used by nerve cells to release pain signals. That is a precise target, very different from simply shutting down the whole nervous system. Walk a tropical shoreline and a live cone shell can still look like something small enough to fit in one hand, while related species use harpoon-like teeth to inject venom into fish. Their venoms contain many different peptides, each aimed at particular nerve or muscle targets. Scientists studying those molecules realized one could interrupt pain signaling without acting like an opioid. The catch was delivery. The peptide does not easily cross into the brain from the bloodstream, so the medicine has to be delivered into the fluid around the spinal cord. In 2004, the FDA approved ziconotide for severe chronic pain in patients who need medicine delivered into spinal fluid. The reversal is unusually clean. A molecule evolved to help a slow snail immobilize prey became a non-opioid medicine for pain that other treatments could not control. The powder with no antidote. On American ranches after the Second World War, a white pesticide known as sodium fluoroacetate, or compound 1080, was used against coyotes and other pests. It has little warning smell or taste, but its real trick begins after the body absorbs it. Picture a cell as a small engine cycling the same fuel pathway over and over. Fluoroacetate slips into that pathway because it resembles ordinary acetate. Then, the body converts it into a molecule that blocks a key step in energy production. British biochemist Rudolf Peters called this lethal synthesis in 1952 because metabolism helps create the compound that does the damage. Symptoms can be delayed for tens of minutes or several hours, longer than a feature film, then progress to seizures, heart problems, and death. Secondary poisoning can also occur when scavengers consume poisoned animals. U.S. regulation tightened decades ago. The EPA canceled registered predator control uses in 1972, then approved a tightly restricted livestock protection collar in 1985.
The final twist is inside the name Peters chose. The body does not merely absorb the poison, its own chemistry completes the lethal step. The poison-turned-beauty treatment. Crossed into a funeral meal in Elazels, Belgium in December 1895, where 34 musicians shared raw salted ham. Within 2 days, 23 developed paralysis and 3 died. Bacteriologist Emil van Ermengem investigated the outbreak and linked it to the organism later named Clostridium botulinum and to the toxin it produced. Botulinum toxin blocks acetylcholine, the chemical message nerves release to tell muscles to contract. When that message stops, eye muscles weaken, swallowing fails, and breathing can become impossible even while the patient remains conscious. The scale is hard to miss. One contaminated meal disabled most of a 34-member band.
German physician Justinus Kerner had described similar sausage poisonings decades earlier, but van Ermengem's investigation tied the syndrome to a specific microbe and toxin. Medicine eventually learned to use tiny, controlled doses of that same nerve-blocking effect. U.S. regulators approved botulinum toxin type A for eye muscle disorders in 1989, and Botox Cosmetic followed in 2002. The outcome is almost absurdly inverted. A toxin identified through a fatal food poisoning outbreak became a treatment deliberately injected to weaken selected muscles. That is, 10 substances that never needed a famous victim to earn their reputation. Subscribe for the next list Science Never Lets You Forget.